Plant stress physiology. First of all, what is stress? Any given and optimal condition of any factor essential for its growth will lead to a broad change in physiological process, and due to this plant body will experience tension and this state referred as plant stress. Stress leads to reduction in growth, yield and death of the plant or plant pot. Stress may be caused by two biotic or abiotic factors. This one, the environmental stress has divided two types, biotic and abiotic. Biotic it's allelopathy, pathogenicity like microbes. Abiotic factors as atmospheric and edaphic factors. In atmospheric has divide three types, light, temperature, air pollution. Edaphic, water and salt. Biotic stress. Biotic stress is an adverse condition in which plant cannot sustain its normal growth due to the interaction with deleterious microorganisms like fungi, bacteria, virus, viroids, phytoplasma, and nematodes. They include biotic problems caused by living organisms such as pathogens, nematodes like insect, and others. Biotic factors leads to reduced shoot development, susceptibility to disease, damaged photosystem and reduced photosynthesis, generation of reactive oxygen, inhibit root growth and impaired cell division criteria, reduced plant biomass, hormonal imbalance and accumulation of reactive oxygen effective on agriculture, biodiversity and environment, and then soil fertility and health devorian, nutrition translocation decreases from soil to the plants. Then abiotic stress. Abiotic stress such as low or high temperature, deficient or excessive water, high salinity, heavy metals, or ultraviolet radiations are hostile to plant growth and development, leads to great crop yield penalty worldwide. Abiotic stress conditions such as high light and osmotic stress are known as trigger systematic stress signaling in plants, which leads to stress responses in unexposed distal tissues, resulting in systematic acute accumulation. The first abiotic stress is temperature stress. This temperature stress divided into the effect of temperature that causes high temperature injury, chilling injury, and freezing injury. High temperature injury. Respiration rate increases and photosynthetic rate decrease. Tissues may be recoiled because of the lack of supporting. Then energy denaturation and aggregation of proteins occur, and RUBISO and other enzymes of carbon metabolism are adversely affected. Then change in the viscosity of the lipid components of the cells to amorph fluid state. High temperature reduce the membrane stability of various cellular membrane, causing low of physiological function and cell integrity. Some plants avoid high temperature injury angle and arrangement of the leaves. Thick cuticle and leaf hairs. It improves conductive cooling. Then chilling injury. Injury which causes due to the low but above 0 degrees centigrade temperature is called chilling injury. This chilling temperature are too low for normal growth but not enough for ice formation. Roots are chilled, the plants may be wild. The chilling sensitive physiological functions include inhibition of photosynthetic and reduced carbohydrate translocations, lower transpiration rates, inhibition of protein synthesis, and increased degradation. Then freezing injury occurs at temperatures below the freezing point of water, below 0 degrees Celsius, and primarily the aqueous phase in the apoplast freezes. At least 0 degrees Celsius, there is a phase transitions in water from liquid to solid. Ice crystals develops in the intracellular spaces and cell walls. Large polysaccharides and proteins facilitated ice crystals formations, which are called ice nucleators. The second abiotic stress, light stress. Alter- Alternation in light intensity, whether a deficit or excess, will result in disruption of plant metabolic process. Low light intensity below the compensation point lead to starvation with carbohydrate used as substrate for respirations first and then other source of energy. In high intensity light, photosynthetic performance of plants is depend upon relationship between photoinhibitions of photosynthetic and the degree of chlorophyll destruction. Then third one, wind stress. The excessive water loss through transpiration causes partial or complete closure of the stomata may be insoluble, which will restrict the diffusion of carbon dioxide into the leaves. As a result, there will be decrease in the rate of photosynthetic and growth and yield. Then other one, nutrient stress. If plant takes less than optimum quantity of any essential mineral, then the situation is referred as deficiency, and more than optimum, then situation is referred as toxicity of the mineral. Deficiency of an element result from amounts and concentrations present in the soil from in which they exist and soil pH. Another important one, water stress. The availability of excessive or inadequate supply of water is called the water stress. Loss of target that affect the rate of cell expansion and ultimate cell size affect translocations indirectly by altering the source to sink relationships for assimilates. Changes in structure of macromolecules by removal of water. Inhibition of cell expansions result in slowing of leaf expansion. Then the dehydrations of mesophyll cells inhibit photosynthesis. Then salt stress. It's also one of the edaphic factors in abiotic stress. Affect plants adversely into two ways. High solute content in rooting medium creates water stress by decreasing osmotic potential, and direct toxic effect of concentrations of ions. Halophytes can grow comfortably in saline soils with high salt concentrations. Glycophytes cannot grow in the presence of high concentrations of salt, but manage to grow in saline conditions by adopting some mechanisms as accumulations of sugar in leaves. Then, how to disease control strategies in plants. Mechanical barriers, cork layers, formation of cork cells in areas surrounding penetration point and forming necroid spot and isolations by cork layers. Tylosis, restrict pathogens transport through xylem vessels. Lignitubers, a sheath may from around the invading hyphae, increasing the mechanical strength of the cell walls. Then biochemical resistance, passive already present in plants. Phenolic glucosides, glucosidates like etc. and toxic to potential invaders. And second one, active induced by pathogen invasions like caffeic acid, scopolamine, and orginol. Like hypersensitive response, limited number of cell side pathogen get isolated in localized necrotic tissues molecule mass secondary, and phytoalexins, low metabolic with antimicrobial activity. Thank you.